So in this video we're going to look at another type of cell division called meiosis. So meiosis is a special type of cell division because it is only involved in sexual reproduction. So it's always about the production of gametes, and gametes are sperm and eggs, or if it's in a plant, pollen and ovum, which is a type of egg, but we tend to use the word ovum in a plant. During meiosis, the nucleus actually goes through two nuclear divisions, so one cell will end up becoming 4 cells, but we will halve the amount of DNA. And that means we produce cells that are haploid. So haploid means one of each copy of the chromosomes. So normally our body cells have 2 of each of the chromosomes. That would make them diploid, and all gametes are haploid. This means, therefore, that the cells are not identical to the parent cells. They are also not identical to each other. So now we have a type of cell division that results in genetic variation, and this is going to be really important when we're thinking about species continuity and evolution and why siblings don't look like each other and all those kinds of things. So let's have a look at the process of meiosis. And as I've said, it has 2 nuclear divisions. So we're going to go through PMAT, so prophase, metaphase, anaphase and telophase. We're going to go through those once. So in this first bit here, where we start off, that's probably prophase. It's not a great diagram of prophase. There is metaphase, anaphase is happening here, and telophase where the nucleus is reformed, and then it happens again. So we have PMAT happening a second time. So prophase, metaphase, anaphase and telophase happen again, but this diagram doesn't show all of them. It shows the two daughter cells here, each one undergoing, well, prophase has happened, metaphase has happened, this is anaphase happening here, and then telophase happening here. So this top diagram is how we make sperm. So you'll see 1 cell at the beginning becomes 4 sperm cells at the end. The bottom row here, we have the first nuclear division, then the second nuclear division, and we're actually making egg cells here, and 1 cell does become 4 cells here, but 3 of those cells have kind of lost their cytoplasm because all of the cytoplasm ends up in one of the cells. So the split of the cytoplasm isn't even. So 1 cell becomes the egg cell, and egg cells will have to have lots of cytoplasm in order to provide nutrition for the fertilized egg. So egg cells, although they do make 4, only 1 actually goes on to be productive. So we have meiosis, 2 nuclear divisions making haploid cells that have only 1 copy of each chromosome, and they're used for sexual reproduction. So let's have a look at how to summarize that simply. So we have a parent cell. Chromosomes make identical copies of themselves. That happened with just before meiosis happened, and then the chromosomes pair up together. Sections of DNA get swapped. We'll talk about that in a minute. The chromosome pairs split up, and then the chromosomes themselves divide. So that at the end, instead of having 4 chromosomes, we've only got 2 chromosomes in each cell. So let's have a look a little bit more detail about how we get genetic variation. Now, the first way that we get genetic variation is during prophase 1. So that's at the very beginning of meiosis, when the pairs find each other. So you can see here that the pairs of chromosomes, so in the cell that's going to become a sperm or an egg. We have the chromosome that was originally from dad in blue, the chromosome that was originally from mum in pink. They have found each other, they've paired up, and you can see something odd happening here. They've actually crossed over. And you can see what it looks like in a microscope. So there's a couple of crossing overs here down the microscope, and this is what it would look like diagrammatically. So the chromosome that was originally from mum and the chromosome that were originally from dad, they actually cross over and they actually break the DNA and swap little bits. And this means that each of these chromosomes is now unique. It doesn't look like it originally did, and it's going to go on and make a unique sperm. So this is called crossing over. It allows for genetic variation because every sperm or every egg is going to be completely different to every other one, because this crossing over happens at random. And this is going to be really important for species survival, because if everything is different in terms of the offspring, then we have variation within our species. And then if there are changes, selection pressures, there's potential for a species to survive if individuals can survive. Now, this is one way that we get genetic variation. So let's have a look at the other way. The other way is called independent assortment, and it happens during anaphase 1. So this is the part of cell division where the chromosomes, they found each other, they've lined up on the equator, and now they're being pulled apart. So you can see here in this diagram, metaphase is happening here, and they're showing all 4 options that could happen at once. So metaphase is when they line up on the equator, and you can see here we've got blues and red chromosomes. In this example, we've got a blue on the left and a red on the right, and then red chromosomes here on the left and blue on the right. Now we can get all sorts of different combinations. If we look over here, we've got all the reds on the left and all the blues on the right, and here we've got the middle blue over on the left, and here we've got the top 2 chromosomes have got their blue on the left. So there are 4 options for how you can arrange the pairs. Do we arrange them like this, or do we swap them over like that? And that's completely at random. And that means when you pull the chromosomes apart to different ends of the cell, you're going to end up with a different pattern for the different sperm options or egg options. So you can see the different patterns. So we end up with different options. We could end up with 1 blue, 2 reds, 1 red, 2 blues, or we could end up with all reds, all blues. So there are lots of different combinations, and depending on which chromosome was on the left and which was on the right, we can end up with massive amounts of genetic variation, meaning that all the eggs and the sperm are different again, which again is important for species survival.